This brief presentation will be about inheritance of some traits in humans, not about diseases yet, because if it comes to genetically encoded diseases, the deep interest of humans in understanding of this mechanism gives us a particularly broad range of topics to discuss, so genetic diseases will be a topic for a separate presentation. Now about non-pathological inheritance. The first question is, how actually can we know if something is encoded by one gene? It is not easy. There are many human traits which are believed to be encoded by one gene, but in many cases these are only beliefs. If you look deeper, it occurs that more genes can be engaged or additional factors can have impact on how the trait looks like. So you shouldn't easily believe in what is said in many sources, including your textbook, stating that this or that trait is encoded by a single gene, this variant is dominant, this variant is recessive. Usually it is not fully true. One of such traits, however, which are claimed with a good support to be encoded by one gene only, is presence of freckles. And it is assumed that only one gene is responsible for presence of them, with an allele encoding their presence and another encoding their absence. The dominant one is this encoding for freckles. It should be like that. Another trait which is encoded by a single gene is ability to taste a specific chemical substance called PTC. Some of people can taste it. They feel the bitter taste for that. Some of people will not receive this taste, and it is also encoded by a single gene. But in most cases, what we think to be relatively easy to determine is actually much more complex. And it will be also partially included in this presentation. How much can we trust that what we think to be encoded by some genes is actually affected only by them? One of these things which obviously depend only and exclusively on a possessed genes are blood groups. I'm not sure if you know what actually it means, so I will also explain a mechanism of how blood groups are used and knowledge about them is used by humans. In general, red blood cells tend to have some specific proteins on their surface. These proteins help our immune system to identify if such a red blood cell belongs to our organism. Such a protein is called antigen. And red blood cells can have two kinds of antigens on their surface. Genetic coding of this protein's presence is really easy. It means like, if the cell possesses an allele A, this allele will encode for production of protein A, and this protein will be present on the surface of a red blood cell. In this case, the organism will be aware that this protein is okay and will not attack cells possessing such a protein. If a cell possesses allele B, it will provide presence of protein B on the surface of red blood cell, and then this protein is recognized as its own and will be not attacked by the immune system. There is also a possibility of having the allele which encodes no protein at all. So of course, if it encodes for no protein, no particular protein presence will be encoded by this allele. What is interesting and rather complex in this case is the reciprocal relationship between these three potential alleles. A and B are codominant. What means that if A and B alleles are present together in an organism, they will just encode for presence of both proteins in red blood cells. So none of them will dim presence of another one. But both A and B alleles are dominant to empty allele encoding for no protein. What means that if you see such a situation, if you see the red blood cell equipped only with A protein, you will not know if such a situation is encoded by presence of one allele A and one empty allele, or maybe the person has two A alleles. The effect would be exactly the same. To understand it better, let's see how it looks like. Two potential genotypes can lead to production of A protein only. If a person is homozygotic for A, will produce only A protein and will have so-called A blood group. It means that the red blood cells possess only A protein. If a person is heterozygotic, having one A allele and one empty allele, then the person will still have A blood group because the only protein which is produced is A. Similarly to that, the same situation takes place for B group. It may be homozygotic for B or heterozygotic mixing B allele with the empty one. If it comes to AB group, the only possibility to have such a blood group is to have both A and B alleles. They are codominant, so they will encode for presence of both proteins, and this is what we see as a blood group. And finally, blood group 0 means that the person is homozygotic for empty allele. So no protein will be expressed on the surface of red blood cell. No protein will be typical for this organism's blood. Why is it important? Of course, practical reasons for understanding blood groups are important when we want to make transfusion. But it is also a good example of how some particular traits can be passed to offspring, because analysis of blood groups helps to understand relationships between human individuals. So you can deduce what are potential blood groups of offspring of a couple with a particular set of blood groups, like here. Let's take such an example. We've got a woman with A blood group, and we assume that this woman is heterozygotic, what means that it is only one dominant A allele, and another one is this empty one, recessive allele. Similarly to that, the man in this couple has a heterozygotic B blood group, what means that only one allele encodes for protein B and another one encodes for no protein. If you'd like to see what are potential blood groups of such a couple, you can use exactly the same Punnett square you know already for solving crosses. There are no differences in attacking such a problem. However, notations can be different. I'm personally in favor of this notation. When you get a base in the form of I, the capital I suggests that there is protein produced, while lowercase I is for a recessive allele encoding for no protein. And if you get a capital I stating clearly that this allele is dominant, then in the upper index you state which protein is encoded by this particular allele. So I'm in favor of this way of noting it down. However, in sources you can find other versions of a notation which are also acceptable. Let's try to solve a Punnett square. Such a person will produce two types of gamete, one carrying dominant A allele and another type carrying a recessive allele for no protein. This person will also produce two types of gametes, one carrying the allele for B protein and another carrying allele for no protein. So there are 4 potential different genotypes produced by this couple. 1/4 of their offspring would be heterozygotic for codominant alleles, so they would have both proteins produced on their red blood cells. 1/4 would carry only the allele for B protein, another quarter for A protein only, and finally 1/4 for no protein at all, what means that their blood groups would be like that. AB, B, A, and 0. In this case, each quarter has a different genotype and different phenotype. Similarly to that, you can solve many crosses involving inheritance of blood group, and I will provide some exercises for you to check if you can solve similar problems. By the way, I think it is useful to explain why it is important. In general, red blood cells carrying particular protein are familiar for the organism, so organism will not produce antibodies attacking the protein which originates from the organism. What does it mean? If the organism has red blood cells with protein A, antibodies will be not produced against protein A in this organism. But of course, there will be antibodies against protein B because there is no B protein in red blood cells. So B protein is something foreign and strange for the organism, and it would be attacked. If the person has group B blood, it means that red blood cells contain B antigen on their surface. Then B antigen is self. It is called self. It is well known and shouldn't be attacked. But A protein for such a person is something foreign and would be attacked by antibodies of the immune system. If a person has AB group, then both proteins are present on red blood cells and both are familiar to the organism. So organism will not produce antibodies against any of them. But the situation is opposite in the person with 0 blood group. No particular antibodies are present on red blood cells. So both types of them, A and B, will be attacked by the immune system. And now, if we want to make blood transfusion, we have to take it into account that if we transfuse a blood of improper group, red blood cells will be attacked by the immune system of the recipient. And here, more or less, you've got it explained which blood group can be received by which blood group. Just skip pluses and minuses right now. They will be explained later. Now try to focus only on AB0. 0 blood group can be given to any person because due to the fact that there are no proteins on the surface of these red blood cells, they will be not attacked by any immune system. So each person can receive a 0 blood group and it will be safe. However, if it comes to being a recipient, the 0 blood group is in a huge problem because it can receive only the 0 blood group. Whatever possesses any protein on the blood cells will be attacked by immune system of the 0 blood group person, because the organism of this person is not used to any kind of protein on red blood cells, and any kind of protein will be attacked. The opposite situation is in the person of AB group. Such a person is called universal recipient, because red blood cells of this person possess both A and B antigens. No antibodies will be produced by the organism of such a person, so no kind of red blood cells will be attacked. Whatever is blood group, 0, A, B, or AB, of course, can be received by such a person and will be not attacked. It will be safe. If it comes to A blood group, such a person can receive blood group of A and 0, and a person of B blood group, B and 0. But for A blood group, if blood containing B antigen will be provided to such a person, B would be recognized as a foreign molecule and attacked by the immune system. I know that understanding of that is relatively complex, and I see no point in further analysis due to the fact that you are and should be more interested in a pattern of inheritance in this case than understanding of the practical implications of this knowledge. Anyway, I decided that it could be useful to understand why we care so much for blood groups, because it is important when we want to transfuse blood. Another aspect of blood groups is Rh factor. It is another protein which can be present on the surface of red blood cells. It is absolutely not related to AB0 groups. So it is independent, it is additional information. Such Rh protein can be present or not. This gene has only two alleles, two potential alleles. One of them will encode for protein, and another will not encode for protein. So it is relatively simple how it is encoded. The person having Rh minus blood group, so having no Rh protein, will be a recessive homozygote, because lack of Rh is a recessive trait. You have to be a recessive homozygote to express the recessive trait. If you got a person with Rh plus blood group, the person having Rh factor on red blood surface, there are two possibilities of genotypes. If a person is dominant homozygote, so has two alleles encoding for Rh protein, the protein will be present and we see the Rh plus blood group. It means that Rh protein is just produced. But similarly to that, heterozygote, having one allele for Rh protein and another one for no protein, still will express exactly the same phenotype. The person will be Rh plus because one Rh positive allele is enough to produce a protein, and such a person will be Rh plus. It also has to be taken into account when we are transfusing blood, because if we transfuse Rh positive blood group to the person who is Rh negative, then of course antibodies of Rh minus group will attack red blood cells of Rh plus group, and the transfusion will be not successful. But there is another problem caused by this aspect of blood groups which should be mentioned. This is a problem of so-called Rh incompatibility. It may happen when mother is Rh negative, but father is Rh positive, so they conceived Rh positive baby. And if it is the first pregnancy, there is absolutely no problem, because immune system of mother will not receive signals about which kind of blood group is present in fetus. Cells of a fetus will not come to the mother's blood, so mother's blood will be not informed about the presence of Rh plus protein in the fetus. And the baby develops normally, no problem with that. But when the baby is born, it usually happens that some of baby cells will get in touch with a mother's blood. In this case, Rh protein from a baby will immunize mother against Rh protein. It is like mother's organism gets in touch with Rh protein and recognizes it as something harmful and will produce antibodies against it. Of course, the baby is born at this moment, so no problem, the baby was born safe. But if mother gets into another pregnancy and this second pregnancy is also Rh positive, then we've got a problem, because antibodies from mother's organism can go into the fetus and they will recognize Rh protein as something abnormal and will try to destroy it. In such a case, there is a huge danger of miscarriage. The fetus would be destroyed. However, if we are aware of the genetic background of the issue, the problem can be easily solved. If we know that there is a risk of so-called Rh incompatibility, which in Polish is called konflikt serologiczny, then the mother is given special drugs which will hush mother's immune system for the length of pregnancy and protect the fetus from destruction. This is how knowledge about genetics helps to solve some health problems in this case. We can predict that having Rh negative mother and Rh positive father, the Rh positive baby can be conceived, and if the second pregnancy is also Rh positive, then this is a situation dangerous for the fetus health. And we can prevent this dangerous situation by administering proper drugs in this case. The next issue we have to discuss, the issue which is probably rather obvious for you, is inheritance of sex. You know that the female sex is generally encoded by the presence of two X chromosomes. In this case, we are not showing alleles. On each of X chromosomes there are many genes. However, you can look at how chromosomes are passed to the offspring. If the mother has two X chromosomes in each cell, then to the gamete only one of them can go. But a mother will not produce any other gamete than X carrying. Do you see it? While in case of the man, who always possesses X and Y chromosome, half of gametes will carry X chromosome and half of them will carry Y chromosome. And in the offspring, these gametes will meet each other and fuse to create the first cell of a child. Mother can provide only X to the whole offspring. So actually, you would say that mothers have no impact of the sex of the child which is conceived. This is not fully true, but not from the genetic point of view. If you look only at chromosomes, it is absolutely true. Mother can provide only X, so mother has no impact on the sex of the baby which will be conceived. On the contrary to that, the full work is for father. If father provides the X-carrying gamete, then the girl is conceived. And if father provides Y-carrying gamete, then the boy is conceived. And due to the fact that each of these gametes has the equal chance of being created, the probability of conceiving the baby of each sex is more or less equal. But we have to remember that not all our traits are affected by genes, or not necessarily by one gene only. If it comes to the most visible cases, we can look at identical twins. Their genetic background is exactly the same. They are identical genetic copies of each other. They are clones. And at the moment of birth, they are absolutely identical, but only if it comes to things which depend on genes. All aspects of our look, health, physiology, or personality, which are not dependent on genes directly, but which can be affected by environment, can be different even in identical twins. So what is identical in them are genes, but not necessarily other things. And another thing is that the longer they live, the more changes accumulate in their DNA. Also, the more effect will be of environment, which may shape how they look like. Different diseases, different experiences, different traumas, and finally, The older are twins, the less and less identical they are. There is also a case of something what is called polygenic inheritance, dziedziczenie wielogenowe, and it takes place if a trait is affected by more than one gene. So here we get an example of a relatively complex issue of inheritance of skin color. In a simplified model, you can say that there are 3 genes involved in that, and each of these genes may have 2 alleles, each of these alleles encoding for a pigment or not. So actually, it means that a person may have one of quite different combinations of alleles. There can be a possibility that for all 3 genes, all alleles will encode for a pigment. It means that such a person produces a lot of pigment in the skin and the skin tone will be very, very dark. The opposite situation is when all of these 6 alleles are for no pigment at all. Then the person will not produce any skin pigment and the skin tone will be extremely fair. But there will be also the whole range of potential tones in between. The person may have only one allele encoding for pigment, so the skin will be very light but not fully light. The person may have 2 alleles encoding for pigment, and so on and so on. So actually, what you will get is the full range of skin shades, from very dark to very fair, with many intermediate tones. And what is important, some of them are more likely to happen because there are more combinations leading to such a situation. So the frequency of these tones will be not equal. Extreme tones will be much less frequent than intermediate tones. And you can see it on the graph. The more of darker alleles, the darker is skin. But the most common are intermediate skin shades. The most common are combinations when only half of alleles encodes for pigment. What you can see is that you get more or less bell-shaped curve describing how frequent are particular skin tones. And we say that in general, in case of traits affected by many genes, the trait tends to be continuous. It means that there are no easy to identify, separate, discrete values of the trait, but there are many intermediate variants which are not necessarily easy to distinguish from each other. So this is not like with that only very light and very dark skin. We may have intermediate shades and not always it is easy to tell which of them you deal with. This is typical for the trait affected by many genes together, and the more genes involved, the harder it would be to distinguish separate variants, so the trait gets continuous. In this case, it would be also very, very hard to reveal the genetic background for the exact variant of the trait we deal with. So you look at the person with a particular skin shade, and you cannot say what is a genotype of this person. On the contrary to nice examples of, let's say, a guinea pig fur we observed in the previous presentation, then it was very easy to say what is the genotype of such a guinea pig, because the fur could be only black or white without any intermediate shades. So once more, to summarize, the more genes involved in encoding of a trait, the more continuous becomes the range of trait variants you can get, and the harder is to reveal which genotype hides behind such a phenotype. But there is also another problem, traits which are affected not only by genes. You can look at this experiment performed by some college. Students of a university tried to show how frequent are different heights among students. So you've got girls in white and boys in blue, and you can see that the distribution of different heights resembles the shape shown on the previous diagram. This is bell-shaped curve, what means that the most extreme heights are much less represented than the most intermediate heights. Why? Of course, due to the rule we mentioned previously. So many genes will be involved in determining the height, but in addition to that, not only genes will be responsible for that. What actually could affect the height of a person? Of course, genes of both parents. However, it will be not a one gene for height. Some genes will encode, for instance, how long your bones will grow. Some genes will encode formation of cartilage on bones. Some genes may probably encode how early your moment of puberty will be set, and it usually sets also the different pace of growth for males and females and so on. So in general, different genes can affect how tall you grow. But in addition to that, there will be many environmental factors which can contribute to your height. For instance, condition of mother during pregnancy. When a fetus grows and some aspects of its physiology are determined during pregnancy, nutrients are provided by mother. So what mother eats and the mother's health condition may affect development of fetus and finally its future growth. Diseases of mother, substances present in her organism, drugs used, many toxic factors that could affect mother's organism would also in turn affect the condition of fetus and affect its future height. And also after the baby is born, its nutrition will have impact on how it grows. Illnesses it can be affected with will also determine some aspects of growth. Finally, which height you get will be the overall effect of action of many different factors, only part of them being genetic. In this case, it dims the genetic background work even further. So looking at such kind of trait, you cannot say how much of the trait is determined by gene and how much is due to environment. So sometimes, of course, it will be visible. You can look at the very tall father and his very tall son and say, yes, that's obvious. He got these genes for tall height from his father. Probably it's right, but you never know how much of this tall height was directly encoded by genes from father. And this problem is very famous in genetics. It is called nature versus nurture. In other terms, how much of any trait you can think about is determined by what you can inherit, how much is determined by what happens to you. This is the genetic background. This is environmental background. And if it comes to more complex functions of humans like behavior, this is also the contribution of two different aspects of our background. Nature, or genes encoding, for instance, for our general personality. But nurture is how our environment, the way we are brought and so on, affects our personality and behavior. These issues are very interesting, not only for geneticians, but also for many branches of human biology and psychology, trying to understand how much of what happens to us is affected by genes, how much may be shaped more consciously by environmental factors we provide to the organism. And this is another reason for which humans are interested in understanding the genetic role in determining many aspects of our personality. The more complex is a trait, the harder it is to determine, but it doesn't mean that you cannot try to reveal it. That's all for this presentation. The next one, actually the last one, will be about genetically encoded diseases, both those which are dependent on point mutations in single genes and those which are dependent on chromosomal aberrations. And it will be the end of the genetics chapter.